but we still have that primer sequence right there, that RNA. And so we can't leave that in our DNA because it's going to mess everything up. So another enzyme called DNA polymerase 1, what it's going to do is it's going to cut out, it's going to remove this RNA primer. It's just going to cut it out, just like, take some scissors to it, cut it out, and remove it. But now, we don't have any complementary nucleotides right here to make our double helix. So in addition to cutting out that RNA primer and removing it, DNA polymerase 1 is going to come back, and let's just say, again, ATGGCCA. And so once we've removed that primer, now we've got this gap that we need to fill in. So DNA polymerase 1 is going to come back and add in the complementary nucleotides that were left, or that were being occupied by that RNA primer. So after DNA polymerase makes the copies, it doesn't need the primer anymore. It's already used the primer, it's done replicating, so we can get rid of that RNA primer, because we can't very well leave a chunk of RNA in our DNA. And so afterwards, DNA polymerase 1 is going to come in, cut out that RNA primer, and then put in DNA to fill in the gaps. Yeah, Daniel?
So, DNA, the polymerase 3, that separates the DNA from the RNA? Or the primer?
The DNA polymerase 3, he's the guy that's coming in, and if he sees an A, he's going to put in a T. If he sees a G, he's going to put in a C. That's DNA polymerase. He's actually doing the copying part, the replication part. All these other enzymes and stuff, they're doing stuff before the replication and some stuff after the replication. But the main enzyme involved in DNA synthesis is DNA polymerase 3. It just has these other enzymes that are helping it out in the process. And so DNA polymerase 3 is going to be the guy, making the copy. He's the one that's looking at the strand and then making a new copy based on complementary nucleotides.
Can you just say, in a nutshell, kind of like DNA polymerase is
It's a copy machine.
transcriber?
Yeah.
Okay.
Not transcribing, because when we're transcribing, we're making RNA.
Okay.
Right? But it's the same idea. But think of it as a copy machine. You put in a piece of DNA and you just make a copy of it based on complementarity. And the enzyme that does this is this DNA polymerase 3. Now after DNA polymerase 3 is, replicated and made a new copy of DNA, we have those RNA primers that were there, that were required for DNA polymerase 3 to land in the first place. But we don't need them anymore, so DNA polymerase comes in, cuts out the RNA part, and then replaces it with DNA. Yeah, Debbie?
I was going to say, is there any way you could draw a visual representation of all of it?
Yes, it's on the next slide. I want. Yes.
Is there a DNA polymerase 2?
There is a DNA polymerase 2, and I forget what process it's used in, but it's not something that comes up in this class, so it's not important to you.
What do they use to write the DNA when they drop the primer?
Ah, good question. So when give me a couple slides and we'll get there. How about that? All right. So here is kind of a visualization, and we still haven't talked about the last enzyme, but I'd like to show y'all what we're actually talking about here. So, yes, Okazaki. So the first thing that's going to happen, this helicase enzyme right here, is going to come in, go on this side, it's going to come in and it's going to unwind this double helix, right? So here it is in helical form. Here you can tell the helicase has come in because it's straightened out all the DNA. And then you have these binding proteins that are there making sure that the DNA doesn't wind back up and go back to helical form. Now the next thing is you're going to have, sorry, what?
What were those proteins? Number 2?
Yeah, number 2, those single-stranded binding proteins. They just call them binding proteins, but they bind to the single strand.
And those are not enzymes.
Those are not enzymes, they're just proteins. Which enzymes are types of proteins,
Right.
but a protein isn't necessarily an enzyme. So these are just proteins, which is why they don't end in A-S-E like all the rest of them do. And so helicase is unwinding the DNA. It's using these binding proteins to stabilize it. And then that's when this primase comes in, right here, and it makes this little RNA primer. And it's shown in green because it's RNA and it's not made of DNA like all the rest of the blue is. And so once that primase adds that little primer right here, then that allows for DNA polymerase 3 to come in, bind to the primer, and then start making a copy of the DNA. And then once DNA polymerase 3 has gone all the way down the gene, you're going to have this DNA polymerase 1 come in, which is not shown here, but it should go in this step right here. DNA polymerase 1 is going to cut out that green or that RNA primer and then go back and fill in the gaps left behind with DNA. So all DNA polymerase 1 is removing the RNA and putting DNA back in there. And again, it's all based on complementary nucleotides.
I missed the polymerase 3. I'm sorry.
All right, polymerase 3. So again, polymerase 3 is the guy doing the actual replicating.
Uh-huh.
And so in order for polymerase 3 to do the replicating, it has to be able to bind to a primer because that's how it attaches to the DNA. So once it attaches to the primer, now it's ready to start replicating. So all DNA polymerase is doing is it's looking at your original sequence, it's looking at your single-stranded DNA, and it's just putting in complementary nucleotides. So it would put in CCC and then ATA. That's all it's doing. It's just making a copy of the DNA using the complementary nucleotides.
It would be T's for that polymerase.
Yes,
Because
because
we're making DNA.
all we're doing is making DNA. Now in transcription we were making RNA, which is why the enzyme was called RNA polymerase. And here in DNA replication we're making a copy of DNA, which is why it's called DNA polymerase. So DNA polymerase synthesizes DNA, and RNA polymerase synthesizes RNA. All right? But they're both polymerases. Anyways, so once this primase adds on the primer, and DNA polymerase 3 comes and binds to the primer, it's going to move down the DNA and make a copy of it. Once it finishes making the copy, it doesn't need the primer anymore, because it's already done its job. So another enzyme, DNA polymerase 1, is going to come in, cut out the RNA, and then replace the gaps left behind with DNA. So RNA polymerase would come in, cut out the RNA, and then fill in the gaps like so. So now you've got all of the rest of the DNA that's been made, that's been synthesized, that's been copied, and now you've got this little piece of DNA. So you end up with a short piece of DNA followed by a long piece of DNA. So what you have to do is you have to glue those two pieces of DNA together. And the enzyme that's going to glue those two pieces of DNA together are called ligase. So if you ligate something, you're tying something together, you're tying something off. So in this case, DNA ligase is going to join these two fragments of DNA. And that's going to give us one long, continuous, nice piece of DNA. And then replication's finished.
Gosh.
Oh, just wait, you're going to hate me in 5 minutes. You're going to hate me in 5 minutes.
God, it's just
No,
like
the
one long
process is
step.
finished.
Yes, the
But you
process is finished.
You have a strand of DNA you've separated, and now they both have two strands.
Yeah. All you're doing is making a copy of the DNA.
Okay.
So our DNA, again, we start with a double-stranded. Helicase is going to make them single-stranded, or at least temporarily, right? And then DNA polymerase 3 is going to come in and make the copy. And then polymerase 1 is going to remove the primers and fill in the gaps. And then this ligase is going to act like the glue that joins these fragments of DNA together on the same strand.
And that all happens in mitosis.
No, this is all happening in a phase that's prior to mitosis. And so this, I probably should have led with this, but so we said that the cell cycle regulates when your cell is dividing. It also regulates when your cell is going to make a copy of your DNA. And so just think of this cell cycle as it's a kind of internal clock for the cell. And so there's 4 phases of the cell cycle. This S phase, this is called the synthesis phase. That means we're synthesizing our new copy of DNA. So everything that we're talking about right now is DNA replication. And DNA replication is going to occur in the S phase of the cell cycle. So when a cell knows that it needs to go and divide, what it's going to do is it's going to jump into the cell cycle. And I say jump into, it's not a physical thing. It's not like they're really jumping into a clock. But, you know, it's just a, it's an abstract way of viewing, this internal clock for your cell. Anyways, a cell's going to start at this G1, and then it's going to go to this S phase. This is where it's making a copy of your DNA. And then it'll go through another phase, and then you'll enter into M phase. This is where mitosis is. So remember we said we have to replicate our DNA before we undergo mitosis? The cell cycle helps regulate that process and the timing of that. I think y'all probably already covered cell cycle in lab, right? So this is kind of familiar, hopefully. Okay. All right. Okay. So here we can see kind of a close-up of what's actually going on here. So again, we're starting with our double-stranded DNA. We have helicase come in, unwind the DNA. We have these single-stranded binding proteins, which are not shown, but they're there. And then we have this primase come in and make this green primer right here. Then we have DNA polymerase, again, going through and just adding complementary nucleotides to start your new piece of DNA. So this green right here, this is actually showing you the primer. And you can see that the primer is complementary to the DNA, except that there's U's instead of T's. And so the DNA polymerase is going to start off by binding to this primer. And then when it starts replicating, it's going to look at the template strand, it's going to see a C and put in a G. It's going to see another C, put in another G. It's going to see an A, it's going to put in a T. Until it does that all the way down the length of the DNA. Make sense? Follow so far? Mm-hmm. Okay. Okay, so this is where you're going to hit me. So what we've been talking about is making one long strand of DNA, with the exception of removing that RNA primer and putting it in the DNA. And then we have to glue those two pieces together. Well, these different strands, so remember we have one that's running 5' to 3', and the other one that's running 3' to 5'. So because they run in opposite directions, they're going to be replicated differently. So the one that we've been talking about up until this point is called the leading strand. You only need one primer on the leading strand. Once you have that one primer, DNA polymerase can go all the way down the length of the gene and make one long big strand of new complementary DNA. That's called the leading strand. And the reason why it's capable of making this continuous strand of DNA is because the enzyme DNA polymerase, it can only add onto the DNA at the 3' end. So DNA polymerase, both 3 and 1, are going to only add nucleotides to the 3' end of the growing DNA chain. And so here you can see this. So if you look at the original template strand in blue, this is the 3' to 5', and this is the 5' to 3' strand. So when we generate our new DNA based on the 5' to 3' original strand, our new strand is going to be 3' to 5', right? Because DNA is anti-parallel. And so here's the primer right here, and so DNA polymerase 3 is going to start right here, and it's going to add nucleotides to the 3' end. So you'll never see nucleotides added to the 5' end. So, and this enzyme, this helicase, this is moving this direction. So this DNA polymerase 3 is just kind of following behind helicase, and as the helicase is unwinding the double helix, then DNA polymerase 3 is coming in behind it and adding these nucleotides to make the new strand.
The green part is primase.
The green part is the primer that was left behind by primase. And so the other strand, the one that's running, this original strand is running 3' to 5'. So when we're making our new strand of DNA, we can only add it to the 3' end. And so we add a couple of nucleotides to the 3' end, but helicase is working this way. So you have helicase going this way, and then DNA polymerase going the other way. That's not a very efficient way to do things. So on this lagging strand, we're going to end up generating lots of little segments of DNA. And so that means that we're going to have lots of those RNA primers along the length of it. So you can see the leading strand compared to the lagging strand. The leading strand only needs one RNA primer. And then once that primer's there, DNA polymerase can jump on and just follow helicase down until it finishes copying the DNA. But on the lagging strand, because our new strand that we're making is 5' to 3', we're adding nucleotides in this direction, yet everything else is moving this direction. So what we have to do as as primase comes in, remember the helicase is moving this way, so everything's kind of moving this direction. Now when when primase comes in, on the lagging strand, it's going to make a primer here, and then it's going to jump forward and make a primer here, and then it's going to jump forward and make a primer here. So it's going to make multiple different primers on that lagging strand, which means that when DNA polymerase 3 comes in, it's, again, the whole process is moving this way. So DNA polymerase is having to start here, start at the primer, synthesize DNA this way, and then it jumps backwards, lands on this primer, and then synthesizes DNA this way, and then it'll jump further on the next primer. So it's doing this kind of backwards leapfrog thing instead of going in one direction. And so, the differences between the leading strand and the lagging strand, the leading strand is going to be made continuously, and you only need one primer to do so. But the lagging strand, because based on the directionality of your original template strands, and the fact that you can only add these new nucleotides to the 3' end, is going to dictate that these little pieces of DNA are made individually. And those little pieces of DNA are called Okazaki fragments.
How does it know when to jump?
What? How what?
How does it know when to jump to the next one? Does it go exactly
Because
halfway?
you got to remember, the primers were put on back here because everything's going this way. So as helicase is opening, primase is coming in, putting a primer, putting a primer, putting a primer. So it knows when to stop when it runs in to that next primer, because it's going backwards. It's starting at this primer right here and synthesizing this way, adding to the 3' end. But since everything else is opening up at the other end, it has to jump backwards.
So once it hits the end of that. then
Once it
it's
hits the end and it runs into this next primer, it knows that it's done.
Okay.
And so then it'll leapfrog back and do the same thing. So it's, it's like one of those like 2-step forwards, 1-step back type thing, or vice versa, I don't know. But it's moving behind RNA polymerase. It's just going forward and then back,
Okay.
and then forward and then back, like this.
I just didn't know when.
Whereas the leading strand, it's just going along like this, smooth. And so now that you have this, complete piece of DNA made from the leading strand, and you have all these Okazaki fragments made, that's where this DNA ligase is going to come in. So it's going to do the same thing it did right here, except on the lagging strand, you're going to have different chunks of DNA. So ligase is going to come in, and it's going to join those Okazaki fragments together. In case any of you are curious, the reason why they're called Okazaki fragments is they're named after the scientist who discovered them, who was Japanese and named Okazaki. So,
What is their function? One more time.
their function, all they are, they're just pieces of DNA. But because of the way that the lagging strand is set up, this lagging strand is running 3' to 5', this blue guy right here. And so when you make your new DNA, you have to make it 5' to 3', right? And so when you're making that DNA, those little chunks of DNA in between each RNA primer, that's the Okazaki fragment. It's just DNA. We just give it a fancy name.
Between each primer.
Yes. Yeah, Alexandra?
So all of those things are happening at once, so like the helicase is on
Mhm.
one, okay.
Yeah, it's just like a little factory. So you got all these parts and pieces coming in together. Everyone's got their own little job. And then they all do it kind of simultaneously and seamlessly. And at the end, you end up with your finished product, which is a newly replicated form of DNA.
You were like, I didn't like you on this one.
I know, yeah, the leading strand and the lagging strand, it gets way more complicated. So this, I'm leaving it to you as simply as I can.
Yeah, don't put any of those answers on the test.
Okay, I know we've tried this before.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site.
Don't worry about that.
Oh, okay.
Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA.
So back to your question, Nancy. So you asked, what are we actually replacing the nucleotides with? Well, we're putting them in new nucleotides, because that's what makes up DNA and RNA. And so remember, your nucleotides, you've got your phosphate, your sugar, and your base. And so you're just going to bring in these nucleotides and just attach them to the growing sugar phosphate backbone. So these nucleotides, they're just floating around in the cell. So when DNA replication is happening, they're just taking those free nucleotides. So for instance here, if this is the strand that you're trying to make a copy of, DNA polymerase is going to see that it's an adenine, and so it's going to signal to a thymine nucleotide to come in, and that will attach to the previous sugar molecule, and then they'll form the complex just like regular. So it's the nucleotides that you're adding in order to make this new DNA. Yeah.
What prevents the random nucleotides in the cell from